We just got done talking about excitation. So how do we excite the sarcolemma? We just talked about the steps that lead up to that. So excitation contraction coupling is the next step in skeletal muscle contraction. And this is how we convert, or how the skeletal muscles convert an electrical impulse in the form of an action potential, convert that to the meaningful skeletal muscle contraction. All right, so that's what it means by excitation contraction coupling, the link between excitation and how that gets converted to contraction. All right, so remember that end plate potential, those ligand-gated sodium channels open as a result of acetylcholine binding. Sodium rushes in, triggers an end plate or a local depolarization. That, when it reaches a particular value, will open voltage-gated sodium channels in the sarcolemma. And when those voltage-gated sodium channels open, sodium rushes into the sarcoplasm and triggers an action potential. Action potentials propagate. So this depolarization, this rapid change in membrane polarization where the inside of the sarcolemma becomes more positive, that happens down the entire length of the sarcolemma. That's what propagates means. Remember, they have the property of conductivity. They can conduct electrical impulses down the length of the plasma membrane. So the change in voltage that opens a few voltage-gated sodium channels, then opens the next, and opens the next, and opens the next, and it gets propagated down the entire length of the plasma membrane. Action potentials the action potential along the sarcolemma will lead to calcium getting dumped into the sarcoplasm, and that is what triggers contraction. Calcium is the link between excitation and contraction. So let's talk about those events. So here's a nice diagram. Here's our axon terminal with our vesicles with the acetylcholine. We know all the events, right? Everything that leads to acetylcholine being dumped into the synaptic cleft, diffuses across, binds to these ligand-gated sodium channels. Sodium rushes in, causes an end plate depolarization, an end plate potential, a localized depolarization right here that reaches a threshold value, which then triggers these voltage-gated sodium channels to open. This one opens, rapid and really great influx of sodium, causes depolarization here, which opens this voltage-gated sodium channel. Sodium rushes in, causes a depolarization, which opens this voltage-gated sodium channel, and so on and so forth. That's what it means by propagation. Now, that is an action potential. It happens down the entire length of the sarcolemma, including these tubule invaginations called T-tubules. So when the action potential travels down the T-tubules, or the transverse tubules, what's right next to the T-tubules? What sandwiches them? These enlarged regions of the sarcoplasmic reticulum called terminal cisternae. What does the sarcoplasmic reticulum do? It is a special form of smooth endoplasmic reticulum which stores lots and lots and lots of calcium. The action potential that arrives at this T-tubule causes voltage-gated calcium channels in the sarcoplasmic reticulum to open. What does the sarcoplasmic reticulum do? Stores calcium. So there's more calcium in the sarcoplasmic reticulum, and remember, in the cytoplasm, it's very low calcium concentrations. I talked about that in the previous video. So when these voltage-gated calcium channels open, calcium rushes out to the cytosol, to the sarcoplasm, okay? And that is what triggers this rapid release of calcium into the sarcoplasm, the cytosol of the skeletal muscle cell. That is what triggers contraction. So let's watch this video.
Stimulation at the neuromuscular junction results in an end plate potential, which then triggers the steps necessary for contraction to occur. We will now focus on this process, known as excitation contraction coupling. Excitation contraction coupling begins with the end plate potential depolarization spreading to the adjacent sarcolemma. When a threshold level of depolarization is reached, voltage-gated sodium ion channels open and allow sodium ions to enter the muscle fiber.
So I'm going to rewind and show you. I want you to pay attention that these negatives and positives are flipping. That is what depolarization is. Remember, resting membrane potential, the inside of the plasma membrane is negative and the outside is positive. But during an action potential, during depolarization of an action potential, we have a rapid change in membrane polarization, and depolarization means that the inside of the membrane becomes more positive or less negative. So I like that it's showing the flip of the charges.
The adjacent sarcolemma. When a threshold level of depolarization is reached, voltage-gated sodium ion channels open and allow sodium ions to enter the muscle fiber. The resulting action potential temporarily reverses the polarity of the membrane, with the inside becoming positive with respect to the outside. The action potential is quickly propagated like a wave. It depolarizes the sarcolemma and spreads down the transverse tubules or T-tubules. T-tubules are extensions of the sarcolemma that dive deep into the muscle fiber. Terminal cisternae from the sarcoplasmic reticulum are on each side of a T-tubule, and together they form a structure known as the triad. When the depolarization arrives, voltage-gated proteins in the T-tubule change their shape. This in turn causes calcium ion channels on the sarcoplasmic reticulum to open and allows large amounts of stored calcium ions to be released into the cytosol of the muscle fiber.
All right, so that is our link between excitation and contraction. It's calcium. Calcium is what will trigger the contraction. So the channels that open in the sarcolemma surrounding the motor end plate and generates an action potential are, all right, so remember, the channels that are in the motor end plate are ligand gated because they bind acetylcholine. The other channels adjacent to the motor end plate are voltage gated because it's that change in end plate potential, the change in voltage that opens them. So the channels that open in the sarcolemma surrounding the motor end plate and generate the action potential, voltage gated sodium channels. So voltage gated. The term propagate, when referring to an action potential, means, so propagate, remember, means it is conducted down the entire length of the sarcolemma. That means spread. It doesn't increase in magnitude. It doesn't get stronger. I didn't say anything about it getting stronger. It just spreads. Okay? That's what propagate means. It just spreads. In order to trigger a muscle contraction, an action potential must reach the blank. Well, we haven't talked about the sarcomeres yet in relation to muscle contraction. Mitochondria does not have, it supplies ATP for contraction, but it does not help trigger, or it doesn't, do anything specifically to contraction. It just supplies energy. I haven't talked about the nuclei. It's the triad. So remember, the action potential has to reach the triad in order to trigger muscle contraction. And it does that because what is a triad? It's the T-tubule, which is an extension of the sarcoplasmic reticulum. The T-tubule, which is the invagination of the sarcolemma. It's the plasma membrane. And right lateral to it, sandwiching that T-tubule on both sides, are the enlarged regions of the sarcoplasmic reticulum. Together, those structures, sarcoplasmic terminal cisternae of the sarcoplasmic reticulum, T-tubule, and then another terminal cisternae of the sarcoplasmic reticulum, that is referred to as the triad. So in order to trigger a muscle contraction, an action potential must reach the triad. A triad consists of, I just talked about this, it consists of two terminal cisternae, which are structures of the sarcoplasmic reticulum, and a T-tubule, which is an extension of the sarcolemma. It's an extension of the skeletal muscle plasma membrane. Blank is released from the sarcoplasmic reticulum, the SR, in response to an arrival of an action potential. So when that action potential arrives at the T-tubule, that change in voltage opens voltage-gated calcium channels on the sarcoplasmic reticulum, specifically the terminal cisternae of the sarcoplasmic reticulum, and that causes the release of calcium into the cytoplasm. Calcium. So released from the SR, calcium, because that's what the SR does. The sarcoplasmic reticulum stores lots and lots of calcium. So as soon as those voltage-gated calcium channels are opened, calcium is going to follow its concentration gradient and rush out to the cytoplasm.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber.
So think of transverse, right? What's a transverse section? It's kind of like a transverse or a horizontal section. It's a cross section. That's why it's called those are called transverse T-tubules. So I'm just going to rewind just a little bit. Here's our here's our sarcolemma, the plasma membrane of the skeletal muscle cell. It invaginates into these tubules, and those tubules just surround the myofibrils. And then also surrounding all of those myofibrils are that blue stuff, which is the sarcoplasmic reticulum. Structure, function, how these are structured, where they're located has to do with the function that it carries out, which is involved in muscle contraction. We want all these structures located near the myofibrils because it's those myofibrils which, in response to calcium, will contract. Sorry. We'll just watch this over again. Sorry.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release.
As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So what is the link between excitation and contraction? It's calcium, right? So one of the many reasons why calcium is so important in maintaining homeostasis, when we talked about the in the integumentary system and how keratinocytes, they synthesize a precursor to vitamin D. Why is vitamin D important? You can't absorb calcium from your diet unless you have vitamin D. What is calcium? Why do we need calcium absorbed in our intestines? It's directly involved in bone health because it's a big component of the extracellular matrix of bone. And then I mentioned several times that calcium is also vital to proper muscle physiology as well as nervous system physiology. And you've seen that because of calcium is required for exocytosis of neurotransmitters. Calcium is also required for contraction of muscles. So calcium is very important physiologically.